growth is measurable. Here the growth is generally increase in the protoplasm, but the here measuring of protoplasm, it is very difficult. That's why here we are using different parameters to measure the growth. Here these are the parameters. Here increase in the fresh weight and dry weight. Here without water, it is the total dry mass is called as dry weight. With water, it is fresh weight. So increase in the weight and increase in the length. Here the pollen tube. So during the pollination and pollination, the pollen grain, it is germinating on the stigma. So here it is germinating. It is also one growth. It is lengthwise, increase in the length and increase in the surface area. The flat organs like leaves, the leaves are also showing the growth. Here like leaves, dorsiventral leaf or isobilateral leaf are leaves showing the growths. Next, increase in the volume. Here the fruits and storage organs. When the here this type of organs, when they are dipped in the water level is increasing the level. So by increase the volume, here we can measure the growth. And here increase in the cell number. Here the one maize root apical meristem, single maize root apical meristem, it is producing more than 17,500 cells, new cells per hour. So it is in the increase in the cell number. Next, increase in the cell size. So the cell size is increasing up to 3,50,000 times in watermelon plant. So here in different parameters, by using these different parameters, we can measure the growth. It is growth is measurable.